Oncogenes are genes that code for proteins that normally direct cell growth. They start out as proto-oncogenes, and then something happens to convert that proto-oncogene into a full-blown oncogene. In essence, some sort of tumor-inducing agent, or it could also be totally spontaneous. Now, proto-oncogenes code for proteins that help to regulate cell growth and differentiation, which makes sense since the essence of a tumor, whether it is a cancerous or a benign one, is unregulated cell growth. In fact, the words oncogene and oncology, which is the study of cancer, share the same root word, onkos, which is Greek for mass or bulk. Now, the products of these genes are often involved in signal transduction and the execution of mitogenic signals. And you should recall that a mitogen is a chemical substance that encourages a cell to start cell division, basically something that triggers mitosis. So how does a proto-oncogene make that switch into an oncogene? Well, there are three main mechanisms: deletion or point mutation, gene amplification or increased mRNA stability, and chromosomal rearrangement. So let's start with the first one. Deletion or point mutation encodes its activity in the cell can lead to a normal protein that is overexpressed. And then finally, chromosomal rearrangement involves translocation of a gene to a nearby regulatory sequence that then causes this normal protein, the gene product, to be overexpressed. Or you could possibly have fusion to an actively transcribed gene which overexpresses the fusion protein or leads to a hyperactive fusion protein. And so really you can start to see a theme here emerging where the key idea is that you have either a normal protein that is just overexpressed, basically too much of the normal protein, or you can have normal expression, but the protein itself is just a hyperactive one. Now, there are tons of examples of oncogenes, including the Src oncogene, RAS, MYC, receptor tyrosine kinase, and also cytoplasmic tyrosine kinases. So let's talk about each one of those here. Src was the first confirmed oncogene, which was discovered in 1970 and was termed Src for sarcoma, which is a tumor of mesenchymal cells or connective tissue cells. It was actually an oncogene discovered in a chicken retrovirus. And so Src codes for a non-receptor protein tyrosine kinase, so it phosphorylates specific tyrosine residues in other proteins. Another example of an oncogene is the RAS oncogene, which codes for a small GTPase, which hydrolyzes GTP into GDP and phosphate. This protein is activated by growth factor signaling and functions like a binary switch, sort of an on-off switch in growth signaling pathways. Examples of downstream effectors of RAS includes the protein MAPK, which is a type of kinase that in turn regulates genes that mediate cell proliferation. You can see examples of RAS oncogene mutations in thyroid tumors, certain leukemias, and cancers of the pancreas and colon. The MYC oncogene codes for a transcription factor that induces cell proliferation, and there's a very common translocation involving MYC between chromosomes 8, where the MYC oncogene is found, and 14, which leads to a certain type of lymphoma called Burkitt's lymphoma. The RTK oncogene stands for receptor tyrosine kinase, which is responsible for adding phosphate groups to other proteins to turn them on or off. These are similar to the Src gene, which is a non-receptor tyrosine kinase, the difference being the location of the protein, either at the cell surface as a receptor or not as a receptor. So say you have a cell surface receptor that receives a signal from outside of the cell. Well, then that signal gets propagated or transmitted into the cell via these receptor tyrosine kinases that add phosphate groups to the target protein, specifically on tyrosine residues. And they can cause cancer by turning a receptor constitutively or permanently on in the absence of signals from outside the cell. Some well-known examples of receptor tyrosine kinases are vascular endothelial growth factor, or VEGF, epidermal growth factor, or EGFR, and platelet-derived growth factor, PDGF. And finally, we have the cytoplasmic tyrosine kinases, which mediate responses to the activation of receptors of cell proliferation, migration, differentiation, and survival. A well-known example is the BCR-ABL gene in chronic myelogenous leukemia, or CML, also known as the Philadelphia chromosome. It is a fusion of parts of DNA from chromosome 22 and chromosome 9. So basically, a part of chromosome 22, which contains the BCR gene, fuses with a fragment from chromosome 9, which contains the ABL gene. When these two chromosome fragments fuse, the genes also fuse, creating this new gene, the BCR-ABL gene. The fused gene codes for a protein that displays high protein tyrosine kinase activity, which is actually due to the ABL half of the protein. And the unregulated expression of this protein activates other proteins that are involved in cell cycle and cell division, which causes a cell to grow and divide uncontrollably, basically becoming cancerous. So as a result, the Philadelphia chromosome is associated with chronic myelogenous leukemia, a certain type of leukemia, as I mentioned before, as well as other forms of leukemias, which are cancers of white blood cells.